This is episode 96 of the Immunology Podcast, Mentorship in Science with Dr. Nicole Quinn. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Round. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Nicole Quinn from STEMCELL Technologies on the podcast to talk about mentorship in the life sciences. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
registration is open for Immunology 2025. Connect and collaborate in Honolulu, Hawaii from May 3 through 7, 2025. Advance your career and access cutting-edge research. There has never been a more important time for the immunology community to gather at the world's leading annual all-immunology meeting at one of pretty much the coolest places on Earth. So learn more at immunology2025.aai.org. Hey, Jason, Happy New Year. It's been a while.
it has been. It's been a few weeks of time off. Well, at least probably.
Yeah. And what a way to start the new year with episode 96, the magical number of wells in a plate. Yeah, I know you don't remember those anymore, but I still work.
I do. I'm validating a plate reader right now.
Fun.
Yeah.
So, are you ready for another year? Did you do something fun over the holidays?
I stayed at my sister's-in-law's house with 10 other people with no door on my room and had to work the whole time.
Wow, that sounds great, Jason. Ask what I did.
What did you do?
Oh, I'm so happy you asked. I witnessed the magical nights of the Arctic, and I saw the Northern Lights in Norway.
What parallel were you at?
Almost 70, 69 point something.
Nice.
Yeah. It was pretty impressive. Pretty impressive.
Pretty cool.
So that was really nice. Yeah.
Sounds significantly more fun than my
just yeah. Well, one of us has to have some fun.
Yeah, it's not me anymore.
Yeah. Well, it's important to start off the year, well. I hope you have many plans for this year. And we're gonna meet soon. So it's gonna be a great 2025.
We do have some conferences coming up, which will be fun.
Absolutely. All right. Talking about good plans. Let's talk some science.
So
what you got?
I got one paper here. It was actually a great plan. A lot of this paper was planning, of all things. It was less immunology than I wanted, but sometimes that happens. Just like sometimes there's more immunology than you anticipate in life. This one, I thought there was going to be more based on the abstract, and there was less. And that's still fine. But so we're going to do a little neuroscience today. So it's in Nature. It's brain-wide cell type specific transcriptomic signatures of healthy aging in mice. First author is Kelly Jin. Corresponding last authors are Bosil Jacques Tasik and Hongqiu Zhang. Came out January 1st in Nature. So they basically did quite extensive single-cell RNA-seq in brains of mice, which you're like, okay, why? But what they were doing is they're looking at very different subsegmental regions and trying to understand what happens with aging over time. And then they used some sophisticated mechanisms to see what was really age associated by region or not in region and all the cell types involved. So there's actually a ton of work. And as I said, a ton of planning involved. And the study is very comprehensive and I think super useful from that perspective. It talks a lot about neuronal things, which I am not going to super deep dive into, which is why this is going to be a little shorter than normals because it's more neurology, some vascular stuff. But there is an immune component because I wouldn't have been talking about it all if the abstract hadn't had some interesting immune component. So they actually find especially in the ventricles, but throughout the immune system, changes in microglia, which we acknowledge as our immune cells, and associated macrophages as well, brain-associated macrophages. And they find that there is a general increase in inflammatory signaling. Notable genes, ILDR2, CCL3, CCL4, UPK1B, So there is this general signaling. They see cytokine production signaling enhance, NF-kappa B signaling, um, MHC1 protein complexes, other stuff. So they see this, and then they see it kind of everywhere in the brain, but in particular the ventricles, which seems to be, especially ventricle 3, which seems to be a hot spot of aging in general. So where they see other biggest changes in aging in a normal mouse, they see even more of this immune component. And then they look at some other downstream effects, and if they're seeing crosstalk of the cells, and they see that too. But fundamentally, it seems, and this is what I think is interesting, which is why it made it on the talk, although not quite as much as I was hoping from an immunology perspective, is that as our brains age, they become more inflamed, which is interesting because a lot of other places our immune systems start kind of giving up the ghost as we get older, but not here. Here, it's an inflammatory party. So that was interesting. And that's pretty much all I got, is that our brain, and they see this overall, and there's upregulation of MHC2 as well in neurons.
In neurons, actual neuronal cells.
Yep, GABA neurons.
So this is in the context of healthy aging.
Yeah, just regular mice getting old, living their best life in a cage.
Okay.
And they just get more inflamed.
So I'm not that surprised by that. Isn't that?
No,
Well,
they don't have any pathogens they're being exposed to, right? These are SPF, these are conventional SPF mice raised, right? So they're not, they may have commensals, but they're not being exposed to the wild environment like you and I are. And usually your immune system becomes more senescent over time, but not here.
And this is just this microglia populations. Is there any kind of infiltration by lymphocytes or other things that they notice?
They couldn't get into that a lot. They see the associated macrophages.
Because one of the things that you to see in general in aging is when it comes to the T cell, the B cell compartments and the adaptive compartment, you see clearly a shift from a more naive, from a population dominated by naive cells to more memory cells. You have all these, particularly in people that are having this chronic viral infections, that CMV, things like that, they really drive inflammation and as seen as a kind of change in the composition of particularly like T cells is impressive, like the difference, especially after 40, 60, most of the cells end up being, kind of these kind of memory cells, many of them CMV uh, specific if you have the infection. So that seems like a temporal, like a very clear source of this immune activation is the accumulation of these cells in time, which is just something that just happens. But that is not what they see necessarily in this brain samples.
Yeah, no, they see this MHC up in the neurons, so maybe that MHC2 is driving other inflammation, but it's just like it's all RNA-seq, right? So they don't have a lot of other stuff going on, but they see just this rising immune signaling, and it's kind of weird.
All right. Well, I guess that's how it is. So, do I have a segue? I don't have a segue, but that's okay because I have such a cool paper that segues are not necessary. Um, so my first contribution to this year's um a little journal club where we have here. This paper, I thought it was really all of it was very interesting, but this one I really liked. Paper is titled "Adaptive T cell Therapy Targeting an Inducible and Broadly Shared Product of Aberrant mRNA Translation." And you know, I am a little bit biased with this paper because this is a paper that comes from my previous employer from the Netherlands Cancer Institute. And it's part of it's a really nice second part of a story they've been working on for some time already. Um, the first author, let me just give the credit where credit's due. So, first author is Julien Champagne from the lab of Ruben Agami at the NKI, and also corresponding author Joanna Oleus at Norway University of Oslo. And just some background here. This was paper published in Immunity, so really nice journal. The story here starts with an observation from this lab that they did some years ago, in which they show that cancer cells, there's a specific type of neoepitope that is appears in certain situations in cancer cells that are known as tryptophan to phenylalanine substitutions. And these substitutions happen under a situation in which cancer cells are being, for example, recognized by T cells, and there's interferon gamma being produced. And interferon gamma can affect a lot of things in cancer. Interferon gamma on its own can be detrimental to cancer, so to the growth and the proliferation of a cancer cell. So that's one of the mechanisms directly. The signaling from the interferon gamma can affect the growth of a cancer cell. And one of the ways is through the induction of the expression of endo endolamine 2,3 dioxygenase 1, so IDO1 for friends. And this enzyme catabolizes tryptophans, metabolizes tryptophan, and therefore generates an intracellular tryptophan shortage. And tryptophan being an amino acid important for protein translation, the idea is that by capturing or by reducing the availability of this amino acid, you can actually kind of interfere with protein translation in tumor cells, and this will help you reduce their proliferation. However, there is a mechanism through which cancer cells, tumor cells, can bypass this particular issue. And there is this thing in which there's this mechanism known as ribosomal frame shifting, in which the phenylalanine is replaced by this this tryptophan, where a tryptophan should have been, is replaced with a phenylalanine. And then it's just in this way, cancer cells can still translate the protein in frame, but just replacing they have they have an enzyme that will bind this phenylalanine to the transfer RNA, and then this can replace the missing tryptophan. And therefore, you'd still have a mostly functional protein that, instead of having tryptophan, has phenylalanine. And so the cancer cell can kind of make do with this. So this is kind of a weird thing, right? A weird adaptation. So one other thing is that in previous work they show that this is something that cancer cells do, and they use this as a mechanism to survive the tryptophan depletion. And so what they set up to do in this work, in a more immunological kind of point of view, is thinking, well, if you end up having all of these proteins that we know from all of our previous work that are going to end up having, this substitution, they are kind of neoepitopes. They are small. They're not mutations in the DNA, but they present as different proteins and potentially can be recognized as foreign by TCRs on T cells. So basically, this is what they set up to find. Can these substitutions generate new antigens that are recognized by T cells? And, kind of spoiler alert, the answer is yes. So what they do is they start, the way they go around doing this, I think, is really nice, because they are quite systematic about it. They look at all the HLA molecules, MHC class 1 molecules, and they're trying to find one in which the consensus sequences tend to have kind of phenylalanine in the consensus sequence, so to increase the chance that such a peptide will be presented. Because otherwise you have so many different potential HLA alleles. So they set out on HLA A24, and they show that indeed they find several peptides that can bind to this. neoepitopes that occur from this substitution, and they end up kind of settling down on one particular neoepitope from a protein that is actually highly expressed on these cancer cells, is TMB-IM6, which is a BAX inhibitor. And this is inhibits BAX-induced apoptosis and linked to ER stress. So this is kind of a protein that is useful for the cancer cell to prevent apoptosis and ER stress. So it's kind of when you would expect it to be highly expressed. So what they see is that under circumstances in which these cancer cells are presented with interferon gamma, they will start kind of, they do this change, they do this substitution, and they express this particular neoepitope. And eventually they find a TCR from T cells, this is all from samples. They find a sample in a TCR that can actually recognize this epitope specifically presented on this HLA, H24, and that it recognizes the neoepitope alone. And that is important in order for these, they kind of do all the controls to show that if you activate the cancer cells with interferon gamma, they upregulate IDO1, they generate this immunoepitope, and then the T cells that are expressing this TCR can recognize and kill these tumor cells. And so basically, you do a lot of tests that show that it's very specific. If you don't have the substitution, then you don't have recognition. And so they think about how can we exploit this for therapy. So you kind of need this tryptophan depletion in order for the whole thing to work. And the main way of doing that is with interferon gamma. So what they suggest is that to have like kind of how would you treat a person or a mouse with this system? So basically they say, well, let's have a two-part approach. If we first target these tumor cells with a TCR that recognizes, for example, a tumor-associated antigen, for example, this MARD1, which is from very highly expressed in melanoma cells, but it has a problem that it's really hard. The toxicity of the TCR, there's a very small window therapeutically, so it's really has not been very advantageous as a therapy to target this particular antigen. But if you put enough kind of cells recognizing this first antigen to generate interferon gamma, to then inhibit tryptophan or to reduce the levels of tryptophan and force the tumor cells to do the substitution, then you can follow up with T cells expressing this other TCR that is recognizing this neoepitope that it comes as a consequence of this. And that combination actually works really great. And I think it's really interesting because then you're very specific. You're really targeting the tumor cells that are expressing some other tumor-associated antigen, but then without the toxicity or the issues that are associated with having to really put high amounts of the T cells recognizing this other antigen that potentially can be expressed in other tissues, which was the issue with the MARD1 in particular. So basically, highlights of this, so kind of in a nutshell, they find these neoepitopes that happen because of this mechanism for the tumor cells to respond to tryptophan depletion. And this can potentially be targeted for therapy because they're only expressed on tumors, and like fibroblasts, normal cells don't do this. So it's very specific for things that happen in kind of dysregulated tumors that use this mechanism to survive tryptophan depletion.
It's interesting that biology finds a way even when it's supposed to, run out of amino acid and kick over.
Yeah.
It's amazing that you can do this, though, and the proteins don't completely come off the rails and break down.
Well, I guess I don't think every protein would survive doing this. I think that as a last-ditch effort for the cancer cell, it makes sense because if the alternative is making no proteins at all, maybe you have proteins that are slightly less functional, but they still do most of the job, I guess. But certainly works. I mean, when this story was initially published some years ago, it was pretty nice. Like, I think people were very surprised. It was pretty cool to see that the substitution was even possible. Um, so that's really interesting. And then, yeah, of course, if you have a different protein, then you have the risk of being detected by the immune system. So there's, in a way, there's no escape.
Yeah, that is interesting. Can't get out, can't get free. There is
Yeah.
no escape from anything. All right. Well, you want also know that there's no escape from when you have me on a podcast?
Entericides.
The gut.
Yeah. All right. What's up with the gut?
All right. Well,
What's your gut feeling today?
this one came from a listener, actually, who had said, hey, I got a paper out, and they messaged in time, right? This is an important thing for people sending in paper recommendations. You can't suggest us cover a paper 6 months old. It doesn't work, right? We try to current things. This just came out, and they sent a message, and it was really cool. And here it is. So, folks, the process that we all talk about at the end of the podcast works.
Yeah, dude, we love getting your emails and your recommendations. So I think that's what Jason said. If you think your paper, is worth talking about, just send us the email as soon as it's out of embargo, so we can read it first thing. If you send it too late, it's too late. We have a policy of fresh only, fresh out of the press. So,
And we
all
have
right.
a little lag over the holidays, right? But it's still fresh.
Yeah.
All right. So here we go. It is InCell, a gut commensal protozoan determines respiratory disease outcomes by shaping pulmonary immunity. First author is Kyle Burrows, and the last author and person who reached out to us is Arthur Mortha. All right, so here we go. Cool paper here, folks. So I like this because it's gut, but it's not just the gut; it's also the lung, and it's eosinophils. And who doesn't love an eosinophil? So there is a protozoa called T-mu, Trichomonas mu something, which I have to go find, but we're going to say it's T-mu, which sounds good, right?
Rolls off the tongue.
Yeah.
Well.
T.mu, not T-E-M-U, the League of Legends character, very different for those nerds who follow.
Or the Chinese online shop.
Exactly, which is spelled the same as the League of Legends character.
All right, come on, give me a science. What's up?
Anyway, so this is known to cause eosinophilia in the gut. It's a commensal. It causes eosinophilia in the gut. What they found was interesting is they see eosinophilia in the lungs, where there is no infection. And so they show these are central circulating eosinophils that migrate to the lungs. So they show that. Okay, so we have eosinophils that are developing and responding and going everywhere. Well, that's weird. Well, what else is happening? Well, then they find out that there's ILC2s, right? ILC2s are, that friendly thing that pump out IL-5 and IL-13 and get the juice going for the eosinophils, right? Well, there's more ILC2s in the gut when you have a T-mu colonization in mice. Okay, cool, right? But, what's really crazy is you also have more ILC2s in the lung where there is no T-mu. But where did these ILC2s come from? So they did some tracing. They are gut ILC2s that go to the lung, where there's no infection. They just get up and walk over, and they bring a whole bunch of eosinophilia with it. Well, that's weird, and it is weird. And they show that they are not crazy, but correct, like any good paper does, right? And then they identify that, the cytokines are important for this, right? But they all the standard ones you'd expect. But they show then it's a tripartite immune network that governs this. You have RAG knockout mice, RAG2 knockout mice don't have the eosinophilia. And it requires both the T cell and the B cell doing different things. And so they do these with these adoptive transfers, transferring different stuff in. And this is all to get to the lung, right? Things behave in the gut. You got ILC2s going gangbuster in the gut without any T cells or B cells, right? We're talking about the lung here. So they can see all the cells clustered together by imaging. And so they figure out that there's an ICOS ligand in B cells that's important. And that is required. So the CD4 cells through OX40 are required for the IL-2 production that is fuel for the fire for the eosinophilia. But then the B cells, I believe, they're involved in the antigen sensing and developing the immune complexes as well. I think I got that right. As I said, they actually map out how each cell does it separately, because what they do is they prime them with that the OTC cells, and so that function was fine. It wasn't an antigen specific for this T cells, which is important. So the T cells effects is an antigen independent OX40 driven IL-2 pathway. So that's just pumping out IL-2 in response to those ILCs doing things, and that's driving the eosinophilia. But the B cells is not the same story for how it's pulling everything together. So then they look at allergic asthma using a dust mite, and they find out the eosinophilia exacerbates allergic asthma, which is not too surprising. If you have a bunch of eosinophils in your lungs, it's a bad party for if you have something in there. And they find that human versions of this bug, similar ones related to TMU, so Dentamoeba fragilis and Pentatrichomonas hominis, are more present in severe asthmatic patients. So they link this in. And then they find that this TMU bolsters early IL-5 dependent containment of tuberculosis. So it's known that people who are dual infected had something weird with tuberculosis. They just have less of this, that this is apparently previously known. And they find that colonization here drives IL-5, which has an early containment on TB. It eventually poops out, but for a while, it holds the TB in place.
Well, but who makes the IL-5 in this?
Well, that's the ILCs.
Okay, the ILCs.
Yeah,
Okay.
long ILC2s, right? Or all the
Okay.
ILC2s.
All right, so it's not because of the eosinophils. The eosinophils are there for basically the same reasons as this.
The ILCs are bringing the ILCs plus the T cells and the B cells are bringing the eosinophils to the party, but the ILCs are also causing a protective effect against TB, but the eosinophils are causing an exacerbating effect against allergic asthma.
Wow, that's a lot.
All from a commensal organism.
That is not colonizing the lung. It's colonizing the gut.
Correct. It's just hanging out in the gut. It's not in the lung.
Yeah. Okay. Just to make
and
that
I think
very
they say
clear.
it's B cell dependent through the ICOS ligand, and that's about it for the B cells. But if you lose B cells or T cells, you don't get the effect. But the T cell spec is antigen independent.
Okay. Very interesting. I mean, these ILCs, they're everywhere. They're just doing all this weird stuff.
Everywhere you didn't know they were and you want to be.
Okay. So to finish our session today, I have a paper which, it's for the textbooks. I think it's a really nice paper about something we thought we understood, but now there's a whole new angle to it. So, just, listen closely. This paper is titled Inflammation Switches the Chemoattractant Requirements for Naive Lymphocyte Entry into Lymph Nodes. First author, Kevin Chen, from corresponding author Jason Sister at University of California, San Francisco, published in Cell as well. And this paper, basically, it starts with, let's say, an observation, which is that when you have, lymph nodes in immune response, you usually have, inflammation, especially if you have a lymph node that is, draining from the point of, infection, or draining from a tumor. You have inflammation, and one of the things that happens in inflamed lymph nodes is that they stop expressing CCL21. And CCL21, as you might remember from the textbooks, is one of the ligands of the chemokine receptor CCR7, which is highly expressed on naive lymphocytes, both CD4, so T cells, and B cells. And there's, this whole spiel we hear from textbook that, CCL21 expressing the lymph nodes is attracting the lymphocytes to the lymph nodes. So because they're expressing CCR7 and CCR7 responds to CCL21. Okay, great. But what happens if you have an inflamed lymph node and it's not making CCL21? Then how do naive lymphocytes get there if all of a sudden the thing that was bringing them there is not there anymore? It's not being produced anymore. And so this is the question. So, and it's very important, right? Because you still want naive lymphocytes to go to inflamed lymph nodes because maybe one of those lymphocytes is what is necessary for this ongoing immune response. So basically what they find is that there is an alternative chemotract and there's an alternative set of molecules that is indeed responsible for recruiting naive lymphocytes into inflamed lymph nodes. And this is very cool. So let's just go a little bit on what are they doing in this paper. Basically, they start showing that indeed, that if you have an inflamed lymph node, you have a downregulation of CCL21. So that's the first thing. They kind of, this is what we know. And particularly, from one of the main producers of CCL21 is the high endothelial venules, which are, important for accessing the lymph node. And so what they find is, although they see a downregulation of CCL21, they see an upregulation of something else, an enzyme called CH25H, which is an enzyme that generates a molecule called 25-hydroxycholesterol. And this enzyme is upregulated in inflamed high endothelial venules. And this oxysterol then is converted to a different oxysterol by another enzyme, which is expressed in the reticular cells. And basically, they generate these enzymes together. They generate these oxysterols, and these oxysterols are the ligands of a different receptor called EBI2. And EBI2 turns out that this is the crucial communication that happens to recruit lymphocytes to inflamed lymph nodes. They show that this upregulation of this oxysterol production and the high expression of EBI2 in B cells and also in T cells, but I think most of this is mostly applicable to B cell behavior, is the way that is a signal that gives naive B cells access to inflamed lymph nodes. And this is something that we did not know before. We were still kind of thinking, well, if there's no CCL21, then who's doing it? And so basically they show using EBI2 knockout mice and a whole set of, experiments with mice that indeed this is the most important part of lymphocyte recruitment into inflamed lymph nodes. Also, interestingly, they show that in an inflamed lymph node, the other ligand of CCR7, which is CCL19, it's either CCL21 or CCL19, which often has been considered kind of redundant because it is for the recruitment of naive lymphocytes. It is kind of redundant. There's no phenotype in the recruitment of naive lymphocytes in CCL19 knockout mice, but it becomes important for recruitment in an inflamed condition when you don't have CCL21 anymore. So it's kind of not as much as EV2, the other receptor, but the CCL21, so CCL19, CCR7 is more important in the context of an inflamed recruitment to an inflamed lymph node than a non-inflamed lymph node. And who is making? So the quick question, okay, so we have these oxysterols that are being produced in the lymph node, and these are attracting the lymphocytes to the inflamed lymph node, but who's making them? So they do a lot of experiment trying to find the source of these um, oxysterol. So, and basically, which is the cell that is upregulating CH25H, which is the kind of the rate limiting enzyme in this. And basically, they show that it is a little, to some extent, high endothelial granules that do produce some, but the main source of this enzyme is Langerhans cells, which are these population, kind of the resident population of myeloid cells. They are radio resistant, and that's how they kind of realize it's them, because they don't, if they irradiate the mice, they still have the cells that are making the CH25H. So that's how kind of they zero down on Langerhans cells that are in the lymph node. And so these are apparently the main source of this enzyme to make this chemoattractant in the inflamed lymph nodes. So I think it's really, really cool because it gives us this answers a very important question. What happens when there's no CCL22, CCL21? And I think it just shows there's a completely new pathway to move the lymphocytes into the lymph nodes that we didn't know before. Isn't that cool?
That is super interesting. I mean, lymph is an underappreciated organ, right?
You think so? Well, I appreciate it very much. Or I mean, it's difficult to do this experiment, right? So they do a lot of like, um, it's quite a tricky thing to study, I think. But yeah, I think I'm surprised we took this long to know this.
There's a lot of things that we don't know until way later than you would think.
Yeah, that's true. That's true. But I also thought I was just saying it was a funny thing that not funny, but I thought it was interesting. They wanted to look into the regulation of CCL21 and why is it being downregulated. And so they wanted to do a tag-seq to look at the locus of CCL21 to see if they see any differences, in epigenetic regulation. And they're like, well, it's actually really hard because CCL21 is part of this massive like gene locus that is full of there's several pseudogenes, several genes all together in the same area. So sometimes I forget how messy sometimes these chemokines and cytokine loci can be. And they actually could not really tell much about epigenetic kind of influence on of the regulation because the locus was so complicated that it was really hard to tell anything. So sometimes you think of genes as this kind of little packages on a chromosome, and sometimes it can be so messy.
Oh, that is very interesting. All right. Well, we're going to be talking to Dr. Nicole Quinn at STEMCELL in just a moment. But before we get to that, we're excited to partner with the International Union of Immunological Societies once again for the IUIS 2025 Congress coming to Vienna, Austria from August 17th to 22nd. Join us for a week dedicated to cutting-edge immunology, innovative research, and global collaboration. Don't miss the chance to be part of the action.
For the second part of our episode today, for our interview, we're really happy to welcome Dr. Nicole Quinn. She's Director of Brand and Scientific Communications at STEMCELL Technologies. And we know Nicole very well. She is working with us at the Immunology Podcast for many years. She's one of the brains behind the whole operation. And today we're going to talk to her about mentoring in life sciences and discuss our sister podcast, Lab Coats and Life, which is focused on topics around mentorship and the life of a scientist. And we're going to talk about her experience running this show and what can we learn from the guests and the topics that have been discussed. So thank you so much, Nicole, for joining us today. I'm so excited to get to talk about the podcast with you and mentoring. I know these are all topics that are very close to your heart. So it's going to be a great discussion today.
Thanks, Brenda. Good to see you. Good to see you, Jason.
Good to see you too. All right. So you have gotten into the podcasting host gig as well in recent years with lab coats and life. So I want to talk about that, the goals of it, what you're planning. But before we get to that, I think there a lot of our guests have thought about doing a podcast or have interest in communication, and listeners are like, oh, maybe I want to do a podcast, or this would be fun. So having gone from helping us produce this podcast to now being in front of a mic, do you have any words of wisdom or thoughts from having a few podcasts under your belt and trying to get in front of the screen instead of behind it?
Yeah, sure. I can talk about that. So, I'll kind of take a step backwards in approaching this question. I left my PhD, not left it. I completed my PhD, but when I did that, I left research with the intention of building a career in science communication. And so, at that time, I thought that I wanted to be a science communicator, the person who actually shared the science. And the way it played out, I really took a sort of behind the scenes role, and I was a producer and developer of scientific communications programs at STEMCELL Technologies. And we can get into what all of those programs are, but one of the outcomes of that program is the Immunology Podcast, and of course the Stem Cell Podcast as well. So I have always wanted to kind of play around with being a science communicator, and it just didn't happen in my main day job. But then we came up with this idea to do Lab Coats in Life as a podcast, and it's not just a podcast, it's also a whole bunch of written resources and toolkits and things you can find online. But we decided to do a podcast to complement those, and we were looking for a host. And of course, we have the four of you. We have Jason and Brenda, and then we have Daylon and Arun from the Stem Cell Podcast as experienced professional hosts. But we wanted, and you all come in and do guest hosting on various episodes, but we wanted a full-time host. And I threw my name into the ring and said, hey, maybe I could do this. Partly because I mean, I'm very passionate about mentorship and all of the topics that we cover. But I also wanted to represent it from a stem cell side, because of stem cell technology side, because of course, all three of these podcasts are produced and owned and put out into the world by STEMCELL Technologies. And that's my employer. So that's how I ended up as co-host or lead host, whatever, of Lab Coats in Life. In terms of doing it, I never wanted to be in front of a camera. I'm very weird and awkward when anyone puts an actual camera. But I do love speaking and public speaking and sort of oral communication. So it kind of was a fit for my background. I've enjoyed it enormously. I love podcasts. I love long form, unscripted, deep conversation. I hate small talk. I'm really bad at it. And so it's a good fit. And I would say, in terms of advice, I mean, it was a good fit for my interests. I don't know if it's a good fit in terms of my skills, but advice for people is, remember there's an edit button. So you can have so many bloopers, and you can edit out those bloopers, and you can try again. And if you've got a good editor, and we have excellent editors on our podcast team, it just makes you sound so smooth and good. And just keep in mind that most podcast hosts are not that smooth. And then just be genuine and be yourself. And if you're engaged in the conversation the whole way through, then that will come through to the listener. So those are my 2 tips.
Those are good tips. I also think, like, I don't know if it happens to you, like we're recording this and we have this conversation, and it's nice. I really like when we have conversation with guests. But it's so strange thinking that this conversation will just go into the ether to some extent, and then you have, like, the thousands of downloads of people that are actually going to listen on to this conversation. And I don't know, for me, it's always so bizarre when I force myself to recognize that that is the ultimate destination of our conversation, is somewhere in the digital airwaves. So I don't know how you feel about that and the permanent record.
Yeah. I try not to think about it. I think that it would influence the conversation, but I know what you mean, because I've been to conferences with the 2 of you, as well as with the host of the Stem Cell Podcast, and I see the interesting reactions when you have people come up to you and say, "Hi, oh my gosh, I recognize your voice," and you. I think it's weird for you. That hasn't happened to me yet. I don't think I'm quite as, I've only done these few episodes, so. But yeah, and then the permanent record, because there's episodes that I feel I was really, I don't know, more articulate in, and then others that I wasn't. And um, I guess I'm more proud of some episodes than others, and wouldn't want to hear some back, and others I'm like, oh, I'm okay with that one. So
Yeah.
yeah,
Yeah. I think that's always the case. Except Jason, he's always
he's always on
he think he's
point.
always great on all of the episodes.
I'm shameless, but which is different than being great. Very different. But the last little thing about being out in the ether, I just thought of and thinking about some of the episodes we've had that I know you've seen live is with AI. Have you ever wondered about what's happening with your voice being out there? I know I've started to think about
Have you wondered about that? I haven't. But I will say, this is probably news to the two of you. We have a tool at STEMCELL where we have played around with cloning your voices, and of course, we will not use them. We will not use them without your permission. But it's really for the point of like covering up bloopers. If you've gave a super articulate, amazing introduction, but you've fumbled one word, we can kind of correct it on the back end. So there's a lot of utility in that voice cloning. We haven't used it yet. We're just
Wait, dear listeners, this is the first time we hear about this live on the show. So we're just learning it right here with you. Okay. Did we sign off our digital identities to stem cell, Jason? I don't remember the
that's
terms of
the
our
ethic.
contract.
They're not going to use it, which is, I think, the point, which is very appreciated, right? They're going to ask for permission, which is cool. That's the point of this, right? It's a bunch of work if I have to go re-record me butchering someone's name for the 50th time, which we know I do very, very well.
Yes. Both of us. Both of us do that, to be honest.
But having this be able to just fix that is, I think, the advantage of this AI, right? Like
I think the line, the very clear, firm, like bright red line in there is, if we are correcting a word you've already said, then I think my opinion is that's probably okay. If we are putting words in your mouth and getting you to your voice to say things that you never actually said in the first place, I think that crosses that line, and I would be quite concerned and I think probably anyone whose voice is out there on the records would be quite concerned about that. And that is a line I don't think we would ever cross.
All right. Good to know. You're on the record saying that, okay? So then,
and I'm very comfortable with that record.
All right. But let's just move on a little bit. Let's talk about the real topic in hand. So the podcast is ongoing. The recordings for the second season. So we already are one season in, and we had so many nice conversations. So the four hosts of the podcast, we shared with you conversations with many guests. I talk about making you're using the online presence to do science communication, about the being a postdoc or not doing a postdoc, not doing a postdoc, the advantages. I know that Jason, what were you talking about in your episodes?
The one I remember is the biotech startup and bench to bedside from last year was the one that really stands out to me. I know there's another, but my brain is
it was regulatory requirements and the regulatory landscape and language. The overall theme of Lab Coats and Life is it's meant to be an adjacent podcast to the STEMCELL podcast and the Immunology podcast. Both of those are very focused on research, and you're talking to researchers about their research. You're doing Roundup papers, or you're covering papers in the Roundup, very focused on the science and the outcomes and the methodologies. But we have found over the years of producing both of those, with hundreds and hundreds of episodes and well over millions of downloads combined, actually, that people, they love that, but they're also asking us through comments and through conversations we've had at conferences and various surveys we've done. They want the mentorship as well. And when you do go into those conversations with when you're doing interviews around, what got you here and what advice do you have to scientists, those people pick up on that. That, combined with the success we've had at STEMCELL with some of our mentorship material, we have toolkits for scientists on how to make a great poster or how to network at a conference. Those always tend to get a lot of attention. We decided to do this adjacent podcast around mentorship, and the theme of it is how to help scientists thrive. And the topics we cover have ranged from open science. There's a lot on open data, on open access. We just had a really
Mhm.
interesting conversation. It'll probably be the first episode of season 2 on PubPeer with one of the
Mhm.
co-founders of PubPeer,
Yeah.
Brandon Stell. We talked to the co-founder of BioArchive last
Yeah. Richard
season.
Sever.
Yeah, Richard Sever. And then, so that's one theme, global accessibility, diversity in science. Brenda, you were involved in a couple of those
Mhm.
conversations. Jason, with his expertise in biotech and regulatory, uh, sort of navigating the regulatory landscape. We've talked about that a few times with various people. And the episode Jason's referring to from last year was about taking your research, like a research idea, and commercializing it, and what is
Yeah.
involved in getting funding around that, what are the legal implications, how do you navigate that. So that's another one. And then sort of technology, how to adopt new technologies, what new technologies are available for sharing science. And I guess maybe the fifth theme is just overall mentorship. So how do we help scientists find their place in kind of the ecosystem of science, and what strengths can they pull on, and how can they find training and mentorship. And um, so we've talked about like how to use social media to improve your you know your brand as a scientist.
Yeah. The visibility of your
Yeah.
research. And what I find of the kind of the array of guests, and that kind of really goes with this topic that you say, there's so much diversity in the things that these people are doing, which I think it's part kind of the mentorship, having role models or having diversity in the things that people do is super inspiring, because then not only helps you as a scientist, but also maybe it's an opportunity to hear about people that are adjacent to the research, the pipetting, in fields that are they're really complementary. And it's so nice to hear that.
Overuse the word synergistic,
Well, I
actually.
think
Synergistic,
Yeah.
yeah, exactly. The
Yeah.
synergy.
Yeah.
an important notion is that you hear this, but it doesn't happen, is that there's more to science than what you study. Right. So everyone in grad school and postdocs, like, I am an expert on this molecule or protein, that's science. And as soon as you become faculty, that's not all you're doing anymore, right? And then what we call mentorship is telling people about all the other stuff that they have to do in the field, but often a little bit late, right? Like they're a little behind the 8 ball when we start typically. And I think that's one of the services that the podcast provides, right? Is like, hey, before you're applying for your faculty grant, you may want to know about all of this that's part of science that isn't just immunology or stem cell signaling or what have you. It's an entire ecosystem.
Yeah. Or maybe you don't want to go into the faculty path. Like maybe
Yeah.
there's another path that you haven't thought of because nobody showed it to you, or you didn't even know it existed, that you should be thinking of. And I've been using this term, the science ecosystem or the science machine. I can't find the right term, but what I've been always kind of known since I had to navigate the departure from research myself, but also and throughout my career at STEMCELL, but really has come to light from all of these conversations we've had with lab coats in life, is that we were trained. Everyone is, I think everyone is trained. Like you go through the undergrad and then your graduate studies, and you're like you said, you're trained in your specific area. You're trained how to be a scientist with, like, the scientific method, and you can develop studies, and you understand how to test things against a hypothesis, and blah blah blah. How to get your science out there through the traditional um, peer reviewed publication system. But that is all changing so fast. And there's so many other things that have to happen and have to be working for all of that to work. And so, although that traditional path, which I think is evolving on its own, is still very much available to people who decide that they want to pursue it, there's just countless other ways that you can contribute to science. And like you said, Jason, you kind of need to know that before you have to know it in order to pursue it. But there is no formal way to learn those things. So that's really the intention behind this podcast.
And it's a struggle. Like I had this problem as a postdoc, being the postdoc president, where we got a combination of people really interested and tons of pushback. No one was in the middle, but the faculty on either side were either very for or very against mentorship programs or career opportunities outside of academia. And some people had the like, well, not everyone goes into academia, and statistically most don't. Another side are like, we're here to do that, and that's our job. But if that doesn't happen, oh well, kind of sides. And it's always interesting because you're right, there's no formal education, and some programs are building it in now as part of something, but it's usually like a course for a semester or a club you can join or something the postdoc association puts on you can go do. But it's not part and parcel, which is interesting because it's very different from engineering, right? If you're in the engineering field, you go and do co-ops. You can take an extra fifth year and go work in industry as part of your degree. The engineering societies are very focused on all careers, not just academic or industry, but it's a mix. So I do think it's something that scientists who are there or who have experienced it. And I know people who've gone to academia from industry and back and forth, but those people who've kind of done it need to advocate for, because it's just going to become a growing, I guess, issue.
Yeah. I would say I see that evolution as well, and I'm seeing it within the societies as well. For example, the International Society for Stem Cell Research, the ISSCR, who we work really, really closely with on the Stem Cell Podcast side, just invited me to speak at a mentorship, like a career mentorship virtual seminar that they're doing. And those types of invitations come around quite a bit now, because they're supplementing the mentorship that the formal institutions may not be providing, or may not just, maybe that's not their role. We still need scientists to be trained in science and in research. And I don't want to put more and more pressure on the institutions to do everything. I think there's
Yeah.
a role for everyone. But the point I would like to make is that traditional path of, the people who are doing the research and putting the research out there, they need all of those, and that's where that word synergy comes in. They need the people who help, like the people who are trained in legal affairs and science, so that they can help navigate the move to commercializing research. And they need the people working in the government around regulatory systems and approvals. And they need the biotech companies who are supplying their reagents. And we need scientists to go into all of these places. And so we're not doing science a service by not giving that information out and helping people get there, and forcing them to figure it out on their own. And then, sorry, the other point I wanted to make is, when people do leave, and I hope this is changing, and they leave that traditional academic path, there can be shame and associated with that. Like, you haven't succeeded because you have decided to move off of the academic train. And I felt it. I had one of my advisors say to me when I said I had gotten this, what I thought at the time was kind of my dream job, because it had science communications in the title. And it was a job out of my PhD, which I really needed. And it turned out to be one of the luckiest things that ever happened to me, is getting this stem cell job. And I had one of my advisors, not my advisor, somebody on my committee say, "Well, I'm really disappointed to see that you're leaving academia." And that, I carry that with me to this day. And
Yeah.
I don't want people to
That's
feel that, Yeah.
it's quite a heavy thing to be kind of confronted with. But well, at least you listened to your gut, and here you are.
Yeah. And to
Yeah.
be fair, I think he was disappointed that a woman was leaving science. It wasn't just about me leaving. And I agree. We don't have good enough representation of women in academia. So
That's fair. I had the interesting thing where my PI vanished and I had to go find a job, and I was given safety nets in academia, but then I landed this job, and now all of the people that were mentoring with me or were friends with me all want to collaborate and ask how they can come join. Some of them ask how they can come join industry, and the rest ask how we can collaborate and what's life like. And it became much more positive, but it kind of was because I had to parachute out, I think, unanticipatedly. And I kind of went all the way just to the cusp of junior faculty, and then pandemic and insanity hit. And so, but yeah, it's interesting talking with the people that were in your orbit, mentors or otherwise, from academia now. I actually have ongoing collaborations with some of them, which is a fun and different.
Right.
Yeah,
We all
yeah.
have a role to play. So, yeah.
Okay, so let's just so last season we the in lab coats and life there was topics included open science, using social media for increasing the impact of your research, new technologies for research, inclusion and diversity in science, the importance of preprints, their mentee mentor relationships. So what are you looking forward now in this upcoming season that will start in March 5th, and with episodes airing every 2 weeks, just like the podcast here? What are you already mentioned Brandon Stell from Papier coming on the show? What else do you want our listeners to know is coming to get them excited about the topics?
So we're halfway through recording. So some of the conversations haven't even happened yet. So I can't really talk about something that hasn't happened in detail, but I will tell you what the topics are. So so we have the regulatory landscape one that we did with Jason a couple of weeks ago. And that interview was with Lynn Santos, who is actually the VP of Vice President of Regulatory Affairs at STEMCELL. And she has decades of experience working in regulatory affairs. And I will tell you that episode, I was playing the role of asking the dumb questions because Jason and Lynn knew so much, and there's so much lingo, and there's an entire vocabulary around regulatory affairs that one has to know in order to pursue this. But I think it's incredibly important because we're seeing so much research accelerate towards the clinic. So that was a really cool conversation. We have one coming up actually with you, Brenda, around global accessibility to science. We have an episode around how scientists are using AI, and specifically not how they're using AI in their research. We've covered that a little bit on the STEMCELL podcast and the Immunology podcast, specifically with that live event we did at the AAI last year. But more how scientists are using AI to supplement their kind of work. So are they using it to write methods sections of papers, or like organize their day, or summarize their thoughts, or come up with hypotheses, or I don't know. We haven't had that conversation yet, but I'm excited about it. We're talking about building a professional brand as a scientist. I think branding yourself is more and more important now that everyone is on the internet, and we talked about the permanent record of somebody's activities and voice. So that'll be interesting. Haven't had that episode yet, but I'm excited about it. And then this is a really interesting one because we talk so much about how to pursue a career, a non-traditional career, like leaving academia. But I don't want to dismiss the importance of staying on that path and actually contributing to scientific discovery from academia. So we're going to talk about how to pursue an academic career and how that has changed over time, and what is different now about academic careers than maybe when all of our PIs and mentors went through. So that one's neat. Oh, we have an episode around learning from failures in the lab. So
Sounds important.
Yes, I think so.
Like useful information.
I think the number one skill any scientist can have is or attribute is resilience and tenacity. So how that comes about, and global scientific funding. So how to access funding no matter where you are around the world, and there's some resources there that we talk about. So those are the ones that the topics we're pursuing, and those all came out of the conversations we had last season. They were just it's easy to come up with ideas for this podcast. So it's the harder part is finding guests, but we've been so fortunate with the guests we have, especially last season when we didn't even have the podcast yet, and we were asking people to come on to a podcast that didn't exist yet, and everyone was so generous. So yeah.
Well, that sounds really nice. That sounds like something to be looking forward to. And well, I did enjoy the conversations I've had so far. Had been really, really interesting. And I'm really looking forward to see the podcast come out. Jason, you have a comment?
I definitely enjoyed nerding out on regulatory things.
Yeah, of course you did. Of course you did.
Lynn and I started talking about strategy after we got off the formal thing about, oh, have you tried this or that? And like, but that's like my day job, right? So
I'm sure Nicole had a blast.
I
But
was
also-
just incredibly impressed at both of them because I think we've talked about this. The production team behind the scenes have talked about that episode specifically because it's quite niche. You have to care about it to be interested in it. And actually, Lynn makes that point in the podcast that, you know, the question was, how do you get into a career in regulatory affairs? And she said the number one criteria is you have to care about it and you have to really have an attention to detail and you want to go through these very boring documents. Well, some people find them very boring, but she doesn't, and then clearly Jason doesn't. I do. You know, I'm a big picture person. I like big picture storytelling. I like what is the implication of all this. I don't want to have to go through the nitty gritty, which is one of the reasons I left the bench. And so I think some people are going to just eat that episode up, and other people might not. But I mean, that's the point, right? Everyone has a different role to play.
Also, the hoop you have to jump through to have a drug, or a product in a drug, which is even if you don't like it, you're stuck with it sometimes.
Yeah. Well, I think that this has been a really cool conversation. Nicole, thank you for coming, talking to us about this our sister podcast and about your interest in mentorship and what is the reason behind this episode and getting this guest in a separate show. And I hope our listeners will tune in to, the first season is already out and on the platform. So if you want to maybe take a listen, and in any case, March 5th will be the first episode we released. So labcoatsandlifepodcast.com. You can find more information, or you can just look for it in, all of the platforms.
And I will say there's also, if you're not into podcast, well, I mean, if you're not into podcasts, you're probably not hearing this. But if you want something in addition to the podcast, if you go to labcoatsandlife.com, you can find all kinds of downloadable resources. You can find episode summaries, transcripts of the episodes, and we're constantly adding more content to that. So you can do that too. All right. Thank you, Brenda and Jason. Right. And I'll see you in a few months. Yeah, we're all going to the AAI, the American Association for Immunology.
Yes, our listeners know.
Yeah, right. Everybody knows this, of course, in Honolulu. So we'll see.
I'm getting my hula hoops and, getting ready.
We'll get you a luau, Brenda, and maybe we'll all go.
Oh, yeah. There you go.
And I hope to meet some of the listeners and come up to me and do that thing that freaks Jason and Brenda out, where you recognize somebody's voice if somebody does.
Scientific Hawaiian shirts exclusively is my new goal.
That's not bad. Can we have like a Hawaiian shirt with this is Immunology Podcast Hawaiian shirt? We should definitely.
That would be very interesting in stem cell orange, a Hawaiian shirt.
Yeah, we'll see. I know the AAI's branding for that specific meeting has been very, very Hawaiian themed, which is kind of fun. So
you have to. All right. So thank you so much, Dr. Nicole Quinn from STEMCELL Technologies, talking to us about the upcoming Lab Coats and Life episodes coming in on March 5th. Thank you so much for joining.
All right. Take care, everyone.
Well, that brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. Or, send a paper away. That's also great. See you next time.